Hi, my name is Kiara Esteves, and this is the work on the prediction of microRNA regulatory mechanisms in Hispanic patients with rheumatoid arthritis. So, rheumatoid arthritis, or RA, is an autoimmune disorder where the synovial joints become inflamed, which limit their function. There are significant health disparities in RA, and Hispanic RA patients specifically experience higher disease activity. However, little is known about the disease in this population because only a small amount of research studies focus on this group. Epigenetics is important in RA because it will activate or deactivate genes without changing the DNA sequence itself. MicroRNAs are non-coding segments of RNA that will bind to mRNA and limit gene expression. We previously identified 13 microRNAs associated with RA in Hispanic patients, which supports the idea that epigenetic mechanisms might play a role in the pathogenesis of RA, specifically in this population. We hypothesized in this study that microRNAs associated with RA in Hispanics will target pathways related to inflammation. To test our hypothesis, we used the 13 previously identified microRNAs and conducted a gene ontology analysis, in which we searched for gene targets in 3 different databases to determine the final list of gene targets. Then, we used David software to identify the relevant pathways in which these would be engaged using the Kyoto Encyclopedia of Genes and Genomes, also known as KEGG. We identified 1,821 gene targets from 3 different databases: TargetScan, MIR-Walk, and MIR-DB. David software identified 15 relevant pathways that would be affected by the identified RA-relevant microRNAs. We highlight here the most likely pathway to be affected, the RAS signaling pathway. This pathway has 39 gene targets with altered functions, indicated by the red stars. Research has found that the RAS signaling pathway regulates T cell proliferation, which is deregulated in RA. We can therefore hypothesize that the previously identified microRNAs alter inflammatory response in RA, but further research is needed to determine the function of these specific microRNAs within the Hispanic patients with RA. In conclusion, we identified 15 relevant pathways that were relevant with the 13 newly discovered microRNAs in the Hispanic patients with rheumatoid arthritis. Thank you everyone, and I hope you have a great day.